Hi guys, my name is Lindsay Miller, and today I'm going to be telling you a little bit my science fair project, which tested the effect of nanosilver particles on the gene expression of Daphnia magna across multiple generations. And a little bit of background, nanosilver particles are microscopic silver particles that are found in many of our common household items, such as food packaging, makeup, and clothing. And recently, scientists discovered that they are being polluted into the bodies of fresh water in a lot of ecosystems. Daphnia magna are a small microcrustacean that test for human environmental tolerance because they share a lot of DNA with humans and many other organisms. So any effects and experiments seen in them can often be applied to other organisms, which is why they are very good for genetic studies. And because this is a fairly new topic, I wanted to see how we as humans are affecting the environment since nanosilver is being polluted into their habitats. So pretty much what I did throughout this experiment was I let them live in a tank containing nanosilver. I had 3 groups, a control group, a low nanosilver group, and a high nanosilver group. And the low nanosilver group was at a 0.1 millimolar concentration, and the high nanosilver group was at a 1 millimolar concentration. And I let them live for 125 days in the tanks. So I checked the population counts 3 times, and that way I could see the mortality rate. Then I used the spectrophotometer to test the bioaccumulation levels within each of the groups. And then after that, I took a DNA sample to be DNA sequenced to see if there was a genetic change in the subjects. So first, as you can see, the mortality rates. So as you can see, I put 50 Daphnia magna in each tank, and at the end of experimentation, the control group saw 113 Daphnia in the tank, the low nanosilver group saw 50 or 90 Daphnia magna in the tank, and the high nanosilver group saw only 24. So therefore, as we can see, at higher concentrations, nanosilver proves to be more toxic because, as you can see, they actually had a death rate in the high nanosilver group versus the other groups had actually an increase, which shows that at lower levels it's not as harmful to them. Then for my bioaccumulation levels, what I did was I used a light spectrophotometer. So pretty much what I was looking for was a major drop between the 380 and 390 wavelength, because that's like nanosilver's most refractive wavelength. So the bigger the drop means that there was a higher concentration at that point. So as you can see, for the high nanosilver group, it dropped from 54.8 to 40.4, whereas the low nanosilver group went from 69.2 to 75.1, and the control group actually stayed from 83.6 to 79.7. So as you can see, the higher nanosilver group had a higher concentration level or higher bioaccumulation levels, so therefore at higher concentrations it can be consumed easily. And lastly, the DNA that I sequenced, I pretty much just highlighted these main sequences that were crucial in these samples, and this is the control group, and this is what was different in the low nanosilver group versus what was different in the high nanosilver group, all these highlighted points. So as we can see, at higher concentrations, nanosilver proves to have a greater or like a higher genetic change on the Daphnia magna. The specific gene that I used was the GAPDH gene, which codes for pigmentation. So this can mean that because that at the high nanosilver group that they had a greater genetic change, that means that at higher concentrations, their genes can be mutated more easily across generations. So these results are very important to understand because we as humans need to understand how we are impacting the environment, and not only how we are impacting it, but we need to try to make a change and try to fix it. So through further experimentation, we can figure out how to extract these nanosilver particles from the water, and maybe even figure out a different product that we could use in manufacturing that would supplement this, so we don't have to use that and affect our environment. So overall, it is just important to understand how we are affecting the environment, because one small change in one organism can offset the entire environmental balance of an ecosystem, and we have to learn how to protect our environment. So I just want to say thank you so much for having me, and thank you.